My name is Roxana Sida, and today I'm going to introduce you through my summer research project, Molecular Recognition of Chemokine Receptor CXCR4. CXCR4 is a receptor of chemokine SDF1, and it's located in the cell membrane. Here is an image of CXCR4 bounded with its chemokine SDF1. We have removed some lipids from the lipid bilayer just to visualize better the receptor. CXCR4 is involved in the regulation processes of proliferation, which is cell reproduction, differentiation, the process by which cells mature to carry out specific functions, cell survivance, and migration. But the most interesting fact of CXCR4 is that when it's overactivated by its chemokine drives cancer metastasis, and it's involved in over 2 dozen types of this disease. Also, it is the co-receptor of HIV. So, the addressing question for our research project is, how does CXCR4 respond to its chemokine? We are interested in identifying unique druggable pockets in the inactive state and possibly therapeutic compounds to inhibit CXCR4 activity. We've been working with three structures of CXCR4: APO, agonist bound, and antagonist bound. We ran 100 nanoseconds of classical molecular dynamics simulations, CMD, for each one of them, and then 200 nanoseconds of accelerated molecular dynamics simulations, AMD, in triplicate. But before going further, I would like to give a brief insight of what is AMD and why do we use it. AMD is an enhanced sampling method that modifies the potential energy landscape by raising energy wells that are below a certain threshold level. So we have the blue curve in our plot that is our energy landscape of the protein, and also we have the dotted line which is the threshold energy defined by us. If at the time we run the simulation the original potential is above the threshold level, it continues its trajectory, but if it's below, then a boost potential is calculated according to a previously defined acceleration factor, which is alpha, and instead of taking more time going through the original potential, we save time by going through a shorter energy landscape. However, if we want to sample the most accurately possible, we must choose a large enough alpha value. The closest it gets to zero, the flatter the new energy landscape will be, and that's not going to be good enough for us to get results of. The next step in our research was doing clustering analysis. That means to minimize data by creating groups of clusters that share similar characteristics. We used the spatial clustering algorithm of Amazon Builder to group our data. The image you see on screen is the principal component analysis plot of the APO structure of CXCR4. The blue dots are the clusters of conformational states that we sample in 600 nanoseconds of simulation. The yellow ones are the top 10 most populated clusters, and the red ones are the states that also represent the conformational space. Since we're trying to make a robust model of transition between activation and inactivation of the structure, we want to explore these states a little more with CMD. Selecting the top 10 most populated clusters might sound trivial, but it was not that straightforward. Here I'm showing you a part of the IPython notebook that was used to get the cluster selections. I modify it from another notebook and make it fit to our system and some specifications as well. And that was the first part of our project. The second part will be to build a markup state model, MSM, for CXCR4. But what is an MSM and why is it so important to build one? Every protein has many states and also many states between those states. However, the most important one that we're interested to find in our system for a longer period of time is the inactive state because, as I mentioned before, we're looking forward to find druggable pockets in the inactive state. And building a markup state model will give us a valuable insight of this. A markup state model is a statistical method that describes the interaction dynamics of interconnected states, assuming that the transition between the states are independent of previous transitions. For example, in the picture we can see all the possible states of protein NTL9, from fully unfolded, which is A, to fully folded, which is N. The arrows are the transitions pathways between states, and the bolder the arrow, the more visited the path is. On the right, we have an example of how a markup state model looks like. The magenta spheres represent the active conformation of a protein, and the blue ones the inactive conformation. Each sphere corresponds to a cluster of similar conformations or a single conformational state. The bigger the sphere, the more populated the state is. And to build a MSM, we just need to follow the methods listed on the left. As you may see, it's all about clustering transitions between states, probabilities, and time.